and start. All right. So as far as labs go, let me see if I can find the plus. So it's above the two dots. Okay. All right. So we've got one new big announcement up here. Our next mock exam, which is on the 28th for you guys, your even day. So 28th, so not quite a week. Obviously, I never see you like exactly a week later because of odd day, even day, except for those weird asynchronous ones. So 6 days from now, when we take our mock exam, you really want to do it on your Chromebook because the only way you can access it, it's through the College Board site, and the only way to access it is through their lockdown browser. Our Chromebooks already have the lockdown browser on there, so all you need to do is like bring it. If you bring your own computer, you first have to install the lockdown browser. There's a 6-page instruction booklet on how to do that. It's in our Google Drive. And then you've installed something on your computer that you may or may not want to have long term anyway. So you definitely want to use your Chromebook. But if you want to install the lockdown browser on your own, that's fine. When you're like, Mr. Cotton, it won't work. I'm like, I don't know. I've never used a Mac or whatever. So if you don't have a Chromebook, you've got about 6 days to get one. The school is trying to give you a free computer. All you have to do is take it. If you're at home, same thing. So our next mock exam has to be taken on a Chromebook. And again, it's not my rule. College Board won't even let you access the test unless you have the lockdown browser on there. The lockdown browser is what we use. ETS, that's who runs College Board AP tests. They're the ones that also run the STAR tests. So like recently, you know how we did STAR tests online? You went to a lockdown browser. Did you guys take a STAR test? History hasn't happened yet, right? So juniors haven't taken one. So like only ninth graders, I think, have taken, ninth and 10th graders are the only ones that have actually taken a STAR test. You know how they've done that on computers? That's through the College Board lockdown browser. So like it's automatically there. Like this is a computer from I don't even know how many years ago, but because it's on the Keller network, it's already installed. Like you open it up and there's this little thing over in the corner that says apps, and that's it. You'll click on that, and that's where you're going to go to take your test. What it does is it just doesn't allow you to leave or go anywhere else. And if you leave, it locks down your test and submits your score. Um, the reason that we want to try one of theirs is, first of all, I've never done it before. I want to see if it actually freaking works. I waited a while, and so this is Mr. Cotton. Mm-hmm, mm-hmm. Um, Today is kind of bad. Is it could you run down the rest of the list, but then come back? If not, I was going to be tearing down lab. Okay, thank you very much. Bye. Okay, so anyway, I want to see if it works. And also, the best prep for like a AP test would be the one provided by the people that write the AP test. So should be good. So what you'll need is a Chromebook, and so that's for everybody. And then if you're at home, you'll also need like your phone on or something so you can be on Zoom. The Chromebook with the lockdown browser won't let you have Zoom on at the same time. So I'd be like on my Chromebook, and my phone would be on Zoom. Again, if you don't want the school Chromebook because you don't like free computers, then you can go to our Google Drive, AP Bio, and there's the page for like how to install the lockdown browser. And there's like a FAQ, there's a troubleshooting page, and everything else. So I don't know how long you've been adulting, but I know chaos when I see it. You don't want to have to install this thing on your own computer. I'm sure it works fine, you know, but so my spidey sense is going off that if you try it, it might not work. So if you're going to do it, you want to try to install it now so that you have 6 days to troubleshoot to get it to work. Um, But just to give you an idea of like kind of what it looks like, um, let me pull it up really fast, what their instructions are. So this is what you would do to install it. Now, part of it is like, do you have this kind of computer or that kind of computer? If you're having Proxer survey trouble, go here and do this. Put in this ID. Make sure you're not on this. Make sure you're on that. Make sure you're in the proxy server and blah, blah, blah. And you're like, that ain't going to work. So I would get a school Chromebook. If there's a school Chromebook that you just borrow from a teacher, I have one. So you can borrow one from a teacher here. So if you're in person and you want to borrow one from a teacher, that's cool. But if you're at home, then you probably need to like get one. So any questions about that? Does everybody have a Chromebook? Okay, cool. I think they were actually like hunting you down, even if you didn't come get one. They like called you down to the library and made you get one. So definitely do that. If you don't have a computer that day, you can't take the test. Like I literally can't give you the test otherwise. If you're at home and you want to come up here and take it on one of our Chromebooks, that's cool. Just let me know. And if you need a Chromebook, right there, Brandy Abbott, our librarian, she checks them out just like books, like really, really expensive books. Okay, any questions on that stuff? Cool. What we're working on today is lab design and standard error of the mean. We actually don't have to solve for standard error of the mean. We just have to know like how to use it. So lab design. Lab design is probably the most important sort of skill type thing that you're going to see for sure. And it's also kind of an Achilles heel for some students because you feel like you already know it. And then you get on the test and you're like, crap, I'm missing something that I feel like I should know from 7th grade. So that's kind of the reason we're going to look at it. So these are some of the things that we want to talk about. All right, let me switch cameras. There we go. So we did go through this maybe in one of the after school review sessions or maybe back with ecology, something like that. We had a question in ecology about like, let's say you have an open field and you add cows to it or something like that, design a controlled experiment. So let's say we'll use a different experiment. Let's say I've got, let's see, give me 1 second. Let me grab something. Okay, so we're going to try an experiment, let's say. And I've got 2 different types of like sanitizer wipes, brand A and brand B. These companies are not very creative in naming their items. But it's easy to remember. It is easy to remember, that's true, and that's supposed to be half of it. Back in the telephone book days, you guys might know this or you might be young enough to where you don't, but companies would name themselves like AAA plumbing, and then you're first. When you look up plumbing, you're ahead. It used to be like A1 plumbing, but If you're like AA1 plumbing, then you're ahead. So there'd be like a company called like AAAAA plumbing, and then there would be a company called like AAA plumbing, and then one like AAA plumbing and A1. Anyway, so I have brand A and brand B, and I want to see which one of these is best. So constants. What are constants? Yeah, all the stuff that's constant that stays the same. Yeah. So everything should be the same. So if I'm going to use a wipe from this one, so if I was going to use a wipe from this one, I want to use a wipe from this one and make sure they're made out of like similar material, same size, and everything else. So constants. If the way I was going to test it, pretend that I was checking for bacterial growth, and these are supposed to kill some sort of bacteria, and I wanted to wipe the bacteria off of pens. Pens. Then I don't know why it came out like that. Then I would wipe like this. I might be like, 1, 2, 3, 4, 5. And then with brand B, I get a brand new one. And it's like, 1, 2, 3, 4, 5. So I make everything the same, everything constant. What's an independent variable? What you're changing. Yeah, what I'm changing. So in this experiment, what's my independent variable? Yeah, the brand. So my independent variable would be brand A versus brand B. Which brand am I using? What would my dependent variable be? The amount of bacteria. Yeah. So I have to somehow measure. Oh, it works on a Mac? Have you installed it? Sorry, quick aside. Niraj, have you installed the lockdown browser on a Mac? Like I know they're in. Oh, go ahead. Uh-huh. Okay, cool. All right, so apparently, like they have directions for how to install it on a Mac and all that. So I know that they say that it works. I was just like, okay. So apparently, we do have one student, Niraj, online that says that he's installed it. He used it in his physics class. Have any of y'all done one of these in physics? Like a normal desktop, and it installed easily? Yeah, it worked fine. Okay, cool. And you can like get rid of it if you want afterwards? I haven't tried to get rid of it. But okay. I imagine I could. Okay, cool. I would think College Board, like it's like state testing and national testing, I would think that it's virus free. But then again, I would think that my bank couldn't get hacked or something like that either. So who knows? So it sounds like on Macs, whether it's a laptop or a full-on desktop, it might be okay. Has anybody else had experience installing this? Which physics was it in physics? Yeah, physics. Which teach? So that was Gallenstein. Okay, is that the same for you, Niraj? Was it Gallenstein's class? Skull? Okay, so Skull and Gallenstein. All right, cool. Thanks. I might hit them up if I have any questions. All right, good. So my dependent variable would be what I measure. So I have to decide what am I going to measure, like quantity of bacteria. So maybe I go along and I get like a swab and I like swab along here. 1, 2, 3, 4, 5. And then I go to a petri dish. 1, 2, 3, 4, 5. Turn it. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. I make everything the same. And so I go along and I swab and I see if any bacteria grows. And then maybe afterwards I count colonies or something like that. So count colonies. Okay, so then what about a control? What would a control be? Do it? Yeah, an unwiped pen. Or if I'm wanting to see if it's like the the chemical that's in this or something, then maybe I get one, like maybe I somehow find one of these cloths that doesn't have the chemical in it. If I'm just trying to test, maybe the only difference between these is like the chemical in brand A and the chemical in brand B, then I would wipe one and it wasn't soaked in any good chemical, it was just soaked in like water. And again, 1, 2, 3, 4, 5. That would be a control. Um, but there's negative and positive controls. Not all experiments have to have both. Negative and positive controls. What you guys normally think of is a negative control. So I get something without my independent variable. What's a positive control? For example, let's say that I try brand A and I try to get some bacteria off of this, and there's 0 bacteria. I try brand B. I treat this. I see if I can get any bacteria off of it. 0 bacteria grows. Then I try just the cloth with water. I try it. No bacteria grows. I need a positive control. What could my positive control be? Is it possible that like maybe there's no bacteria? It could be possible that it's like I might determine that like, oh, it's like the friction or something like that is actually killing the bacteria. But it's also possible that like all my bacteria are dead. So what I would do, or maybe the agar that I'm trying to grow my bacteria on, maybe it's bad. So what I would do is I'd take something like, here's a plate that's growing bacteria. I can see it. I'd scrape some off, put it on my plates. Let's say nothing grows. All right, my plates are bad. So a positive control is kind of the opposite of a negative. That's when you're expecting something to grow on there. We had a plate in our lab that was LB and it was P glow negative. What do I expect to grow on that? 0 stuff or a lot of stuff? A lot of stuff. So when you start looking through like all of our petri dishes, they were all kind of there for a reason, right? So I've got a negative P glow and I put it on the LB ampicillin plate. What would I expect on that one? Nothing to grow. Why? Yeah, there's an antibiotic on there, and it basically should have gotten killed. So I sometimes have to come up with like 2 controls, negative and positive. The default thing that you always think of as a control is typically negative. And then I compare that to my experimental group. My experimental group, again, is like, do I have brand A or brand B? Okay, the next thing, it seems to pop up a lot more recently. Anytime I want to increase reliability, I just increase my sample size. So the more I do the experiment, the more certain I am about my results. So if I do this experiment once and brand A wins, I'm not that confident that brand A is better. If I do it 30 times and brand A wins 30 out of 30 times, I'm pretty sure brand A is better. So increasing sample size increases reliability. This question came up on our first mock exam, I think. There was something on there where it came down to reliability. Reliability is pretty much always, in our situation, going to come down to do the test more. Either do one test with 50 samples or do one at a time 50 times, but do it as much as you can. A bigger sample size is better. A null hypothesis. With a null hypothesis, you always assume there's no difference. So what would my null hypothesis be in this experiment? Yeah, they're the same, and they're the same as what? They're the same as nothing. They're the same as my control. So my null hypothesis would be, hey, whatever happens when I take just a cloth with no chemical on it, however many bacteria grow there, let's say there's 15, then my null hypothesis is that when I treat it with brand A and brand B, it'd be 15. So my null hypothesis is just assume that nothing changes. If I want to test Gatorade and Powerade on my athletes, I'm going to assume that it's no better than just regular old water. That will be my null hypothesis. And then let's say I get a difference. So let's say I was expecting no difference, but then there is a difference. Now I have to use stats to see if it's significant. So about the only statistical test that we've done all the way through is chi-square. There are others if you're in a stats class, but we're not. So if I was going to sit down and actually do some math equations, it'd be chi-square. So I was expecting 10 bacteria. You know, here's my control. It had 10 bacteria grow on it. This one had 8, and this one had 4. So this one's better. But based off of chi-square, is it better enough, or is that probably random chance? This one's better than nothing, but is it better enough, or is that probably just random chance? Chi-square can tell me that. All right, the next thing. Mathematicians wrote it this way. You're like, does that say sex? Kind of. That stands for standard error of the mean. So, mean is that, and standard error of the mean is that. So let me show you what standard error of the mean is. Let's say I've got a question like this one. I'll switch cameras for you guys at home. All right, we're going to try a quick little free response, and it's going to involve standard error of the mean. So I'm just going to kind of throw it at you, see what you think, and then I'll kind of explain standard error of the mean. So back in 2018, question fours were worth 4 points. This year, question 1 and 2 are supposed to be worth 8 to 10 points each. We don't know until we see it. And then question 3, 4, 5, and 6 are all worth 4 points. So 4 of your free response should be similar in length to like this one. So just like always, if I go into Google Drive and I go to Notes, Unit 8, this file is in here. This is 2018 number 4, and it's the 23rd page in the file if you want it and you're at home. Okay, so here's what it says. Number 4, 2018. The common bed bug, that's its genus and species, Cimex, let me give this a shot. Cimex lectularius. That makes sense. Is a species of insect that's becoming increasingly resistant to insecticides. Okay, cool. Bed bugs possess several genes suspected of contributing to the resistance, including P450, ABC8, and CPS. To investigate the role of these genes in insecticide resistance, researchers deleted one or more of these genes in different strains of bedbugs, as indicated in Figure 1, and treated the strains with the insecticide beta-cephalothrin. Each strain was genetically identical except for the deleted genes and was equally fit in the absence of beta-cephalothrin. FL followed by a TH. That's tough. The percent survival of each strain following treatment is shown in Figure 1. Okay, so part A says identify the control strain in the experiment. Identify the control strain in the experiment. So down here, if it's a little bit tough for you to read at home, it says Figure 1, percent survival of 5 strains of bed bugs treated with beta-cephalothrin. A positive indicates the gene is present. It's not president. The gene is present, and a negative indicates the gene is deleted. All right. So now we want to figure out which strain is the control. All right. So for you guys that are in person and I can see your hands, we'll vote. If you're online, you can just put a number in there. I've got strain 1, 2, 3, 4, and 5. All right. If you think that strain 1 is the control, raise your hand. All right. How about 2, 3, 4, 5? Nice showing. It is 1. So the reason that 1 is the control is because that's what's out of nature. All of them have the wild type version. So this is a typical bacteria. So just nothing special. My control is, I guess I didn't write it up there, but a control is something that doesn't get the independent variable. So it has no mutations. All right, next up. It says use the means and confidence intervals in Figure 1 to justify the claim that ABC8 is effective at providing resistance to beta-cephalothrin. ABC8. Okay, so now it's where we get into standard error of the mean and confidence intervals. So I can't remember when, maybe it was an after school review or something like that. but I did mention this once in the past. I'm looking for ABC8, and I'm trying to figure out if it's significantly good at helping the bacteria survive. Okay, so what do you think these confidence intervals mean? You're like, I don't know how confident you are. How does this one compare with this one, with this one, with this one, with this one? Huh? It's accounting for variability within the experiment. So they're like pretty sure that the true value is within. Yep. So we're accounting for variability. So we're pretty sure, kind of like how we can select p-values with chi-square, they'll select the equivalent of a p-value, kind of the same thing in here. So usually they'll have like a 95% confidence interval or something like that. So we're like pretty sure, like 95%, that the real answer is somewhere in there. Okay, what does it mean if I compare this bar to this bar in the graph? What do confidence intervals have to do with comparing those two? Yeah. You can't consider them different because the confidence intervals overlap. So it's probable that they're like identical. Yeah. So statistically, I'll kind of summarize. So statistically, these two are not significantly different from each other. Why? Because that interval and that interval overlap. So when I look at this bar graph and I look at this bar graph, like that one is higher. But statistically, we're like, yeah, we're not that certain that this is actually different from this one. What about this one versus this one? Those are different. Here, these guys do not overlap. So I'm pretty sure that strain 1 really does outsurvive strain 2. I'm not very sure that strain number 1 right here outsurvives strain 4 because these intervals overlap. I am pretty sure that 1 outdoes 2 and 3. And that's what these confidence intervals are. So when they talk about standard error of the mean and overlap, anytime these things overlap, and they've asked some version of this on every free response since they redesigned in 2013. They always ask this. Something to where like getting the answer right means that you go like, oh, that and that, those don't overlap. So yes, statistically, those guys are different. That one and that one, they do overlap. So statistically, column 2 and column 4, they're not statistically different. And you can literally say it like that. Because the intervals don't overlap, 1 and 2 are statistically different from each other, significantly different. Here, there is no significant difference because the error bars overlap or because the intervals overlap, they'll say. So that's how I read error bars. All right, so if I'm trying to justify that ABC8 is effective at providing resistance, how do I do that? I'm a bed bug. ABC8, the claim is that ABC8 helps me survive when I get sprayed with flufloxacin. And thank you guys in the chat, by the way. You were right on voting on column 1. Yeah. When it's deleted, there's a significant lower percent survival. Yeah, when it's deleted, there's a significantly lower survival. Which columns are you comparing? Like, which strains are you comparing? Okay. Which ones? What you said was right, but which ones am I comparing? 1 and 3. Yeah, 1 and 3. So here's 1. 1 has all 3 regular genes. And so the likelihood that they survive is, 70%. Over here, once I have a mutation, if a bedbug has a mutation for ABC8, that's the only difference between column 1 and column 3. And when I do that, that guy is lower than that guy. The bars don't overlap, so there is a significant difference. So it's exactly like you said. But then make sure anytime that you justify something, you have to tell me like how you know it very specifically. Like when I compare strain 1 or column 1 with strain 3 or column 3, here's what I notice. All right, part B. P450 encodes an enzyme that detoxifies insecticides. ABC8 encodes a transporter protein that pumps insecticides out of cells. Most chemotherapy drugs that quit working, the way that they work is one of our natural pumps, kind of like a sodium potassium pump, would mutate and it starts pumping out the chemotherapy drug. And so that's why grandma's tumor was getting smaller and smaller and smaller and smaller. And then all of a sudden the chemotherapy drugs seemed to quit working. And now it's getting bigger and bigger and bigger. So again, P450 encodes an enzyme that detoxifies insecticides. ABC8 encodes a transporter protein that pumps insecticides out of cells. CPS encodes an external structural protein located in the exoskeleton that greatly reduces the absorption of insecticides. Okay, based on this information and the data in figure 1, explain how deletion of both P450 and ABC8 results in lower survival compared with the deletion of CPS only. That's a lot. Explain how deletion of P450 and ABC8 results in a lower survival than with CPS only. And as far as when they say looking at the figure also, it's literally just going to confirm the same thing. Like it's literally going to show that what they said is right. So you're trying to explain why is it that a deletion of P450 and ABC8 helps them, or I'm sorry, hurts them more than a deletion of CPS. Okay, now, this is one of those questions where at the end, after you answer it, you're like, blah, 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 as your answer. You will come across those. Some of them is like, will be like, I feel like I'm restating stuff from the question and I'm just like, here's my evidence. So on this one, um, well, what it comes down to is P450, its job is it detoxifies insecticides. So if insecticide gets into me or not, it detoxifies it. ABC8 transports the stuff out of my cells. So even if it gets into my cells, they can transport it out. CPS is good at like keeping the stuff from getting in, but once it gets in, I'm screwed. So it literally just comes down to like, well, um, the reason why is because 450 detoxifies it if it gets inside. ABC8, if it gets inside, I push it out. And so if it gets inside, I can take care of it. If it gets inside, I can push it out. Here, this only helps me keep it out, but if it's not perfect, then it gets inside and kills me. Like you're literally just sort of like repeating what they told you they did as your evidence. Isn't that lame? And I know this because like I look up. So a lot of these I'll be like, this is the answer, this is the answer, this is the answer, that's cool. I know what kind of things they would want. And then when I come across questions like this, I actually go and I check the rubric because I'm the same way as you. I'm like, this is the answer? And they're like, yes, that is the answer. And you're like, okay. So you'll come across some that just seem sort of dumb. All right, let's try one more. So this is where they'll say something like the nervous system is not on the test. And you're like, that is absolutely the nervous system. And they'll be like, no, it's not. This is a lab question. And I'm like, about the nervous system. So this is classified as a lab question. Okay, this one was 2019, number 4. So this is also a 4-point question. It says this: acetylcholine is a neurotransmitter that can activate an action potential in a postsynaptic neuron. Okay, they label my presynaptic neuron, my postsynaptic neuron, there's the synapse. Okay, great. A researcher is investigating the effect of a particular neurotoxin that causes the amount of acetylcholine released from the presynaptic neurons to increase. So it'll increase the amount of acetylcholine released from a presynaptic neuron. All right, part A. Describe the immediate effect of the neurotoxin on the number of action potentials in the postsynaptic neuron. Predict whether the maximum membrane potential of the postsynaptic neuron will increase, decrease, or stay the same. So we'll just work on part A for now. Describe the immediate effect of the neurotoxin on the number of action potentials in the postsynaptic neuron. This neurotoxin is actually one of our bug killers. So like when you go and you spray some stuff on like an anthill, some versions of that is it makes their acetylcholine like release to the point of death. If you keep telling my diaphragm to contract and it can't relax, then I breathe in and I never breathe out and I die. I suffocate from the lack of the ability to exhale. So contract, relax, contract, relax. Too much acetylcholine, contract, and that's it. It'd be a horrible way to die. All right. So what's the immediate effect on the number of action potentials on the postsynaptic neuron? What do you think? More, less, or the same? Do what? Yeah, more. It increases. And so more acetylcholine means I open up more of these chemically gated ion channels, which means more sodium or whatever goes in. And so now I should have more action potentials. Did it bother you that they said immediate? Like they went out of the way to say immediate, and you're like, well, crap. I mean, like when it releases, it binds. Like, is that immediate enough? Because like then it has to open, and then it has to like let sodium in. Like that bothered me. I thought it was kind of mean. All right, next, predict whether the maximum membrane potential of the postsynaptic neuron will increase, decrease, or stay the same. So what do you think? Up, down, or stay the same? Okay, decrease. I'm not saying you're wrong. There's several students that have their thinking expression on. So I've kind of got you as a tally for decrease. Since there's three options, we can actually vote on this one. I should have voted on the last one. So take a second. Predict whether the maximum membrane potential of the postsynaptic neuron will increase, decrease, or stay the same. And then we'll vote. All right. So if you think it'll go up, raise your hand. Think it'll go down, raise your hand. If you think it'll stay the same, raise your hand. It stays the same. What word allows me to know that it'll stay the same? What important word is in that sentence? Maximum. So it's still going to go up to about plus 30 and then go back down. Now, immediately, the more I open, the faster I'm going to reach threshold. But the maximum membrane potential is going to stay the same. That's why we teach the nervous system, even though they say it's not on here. I would only have a shot at getting that, or I have a much better shot at getting that if I understand the nervous system. Plus, the nervous system's cool. Second, do you realize why this is partly a reading comprehension test? I know it's a reading comprehension test. That's why I got hung up on immediate for a while. If I'm a student, maximum membrane potential. And all of a sudden you're like, oh my God, I missed it because of that. Like I understand this stuff. All right, part B. A researcher proposes two models, A and B, for using acetylcholinesterase, an enzyme that degrades acetylcholine to prevent the effects of the neurotoxin. In model A, acetylcholinesterase is added to the synapse. In model B, acetylcholinesterase is added to the cytoplasm of the postsynaptic cell. Predict the effectiveness of each and provide reasoning. So are they both effective? Is one effective and one's not? And why? Model A, we add acetylcholinesterase to the synapse. Model B, we add acetylcholinesterase to the cytoplasm of the postsynaptic cell. Predict the effectiveness of each and provide reasoning. Since they have each in all caps, I don't just say, this one does this. It's like, what does the other one do? All right, so model A, I add acetylcholinesterase to the synapse. What do you think that will do? Do you think that that will shut down nerve transmission, or do you think it will ramp it up? Okay, okay, shut down. Raise your hand if you think it'll shut down. I agree, it'll shut down. What about if I squirt it into the postsynaptic neuron? No, why not? Why won't it do anything if I squirt acetylcholinesterase in here? Yeah, the receptor's on the outside, and like acetylcholine's not in here. So acetylcholine is in the synapse. So acetylcholinesterase degrades acetylcholine. So putting it in the postsynaptic membrane doesn't do anything because the acetylcholine is out here. So it's only going to have an impact if it's in the synapse. If it's in the postsynaptic neuron, it won't. So one of the things that is hard to kind of replicate is fatigue. This is like question 4. So that means you're like 2 hours into the test. So you've already been thinking hard for 2 hours, and then you come across this, and you have to catch little subtle things like maximum. So I don't really know how to simulate that for you other than like you work hard in this class, then you leave and you probably go to another hard class, and then you probably go to another one. You know how you feel in like 8th period? That's how you'll feel at the end of an AP exam. And then hopefully you don't leave mine to go and take physics or something like that. It used to be that AP Bio was in the morning, and on the morning of the AP Bio physics set, on the day of the morning AP Bio test, the afternoon was physics. And so juniors that were enrolled in both went back to back, AP Bio, AP Physics. You had like a 30-minute lunch break in between, and that was it. And I was so glad that I was first. I used to go and thank the physics teachers. I'm like, I'm so glad I get their brains when they are fresh. So you guys get them, their brains are used, or like new condition, whatever they call it with cars. So, all right. The last thing that you're going to see pretty much for sure is a rate calculation. So rate calculations are something that everybody thinks that they know. But when I ask them on quizzes, about a third of my AP Bio students miss rate calculations. If I ask a rate calculation one, at best, 60, 70% of students get it right, and that's every single year. So make sure you can get the rate calculation. Let's look at an example. Hang on. There we go. All right. So let's try this one. Actually, try the math because I know that you think you can do it, and 2/3 of you can, but 1/3 of you mess it up. So we want to catch that error now instead of on test day. So, and it can be any just scrap piece of paper, you're not turning it in or anything like that. So based on the data shown at the right, calculate the average rate of increase in oxygen consumption for animals acclimated to 5 degrees Celsius as the temperature increases from 10 to 20 Celsius. So average rate of increase in oxygen consumption for animals acclimated to 5 Celsius. Where did Zoom go? Oh, a little bit to the left. There we go. Animals acclimated to 5 Celsius as the temperature increases from 10 to 20 Celsius. All right. Some of you guys are still writing. I'll give you just a little bit longer, and then we'll do our answers. If it's just a whole number, you'll show me. So let's say that you thought the answer was 1, you'd be like this. And if you think it's like 1.5, then just do, like 1.5 or something like that. All right. So on 3, you're going to show me what you think the answer is on your hands. 1, 2, 3. I got 4. So let's walk through it. So acclimated to 5 degrees Celsius. So I've got 2 options. I got the circles, that's 5 degrees Celsius. I got the triangles, that's 25. So I want to follow the circles. Okay, so ignore the triangles. As the temperature increases from 10 Celsius to 20, so my temperature range is here. So I'm going from that dot to that dot. So I want to figure out my rise. If I'm going to show my work, I guess I have to say like, change in y over change in x. So like my y2 minus my y1. So I'd have something like 120 minus 80 equals 40. So I rose that much. From left to right, I went from 10 to 20. So that'd be like 20 minus 10 equals 10. Let me flip back to this. So I'd have something like this. I'd say 4. And even if I don't understand the units, because it's rise over run, it's rate, it's slope of a line, I just take whatever units are on the y-axis and I put them over whatever units are on the x-axis, even if it's a fraction of a fraction of a fraction of a fraction. So I literally am going to write 4 millimeters of O2 per gram per hour per degree Celsius. And you're like, what does that mean? I don't know. There's a whole lot of this for this for this. I mean, I can like this many milliliters for every gram produced per hour for every degree. Like I kind of get it, but like it seems ridiculous. But I just put it there. You guys have the written test, so units count. The written test will be different from the online digital test. We can ask you stuff in person on a written test that we know you can't look up, and it's like, all right, we can ask them that. But we might not ask that question to somebody taking it on a computer because that's an easier question to Google or something like that. So units, don't forget them. All right. Questions so far? Let's look at one more, and then we'll see what we got for TCTV. We'll call it a day. This one showed up on your first quiz or second quiz or something like that, I think. I used this graph. I don't remember which one I asked, but like I used this graph on one of your old quizzes too. All right, so number 10 here. It says the graph at the right depicts the relative activity of a particular enzyme as pH changes. Describe the shift in relative activity between pH 8 and 10 and explain why that occurred. Nowadays, they'd probably say justify instead of explain. Same thing. Describe the shift in relative activity and explain why. So if you're at home and you can't quite read it, this is pH on the x-axis. This is relative activity on the y-axis. All right, so what would I say for describe the relative activity? What are they looking for? Yeah, decreases. If I had enough numbers to where I could like say what the slope is, I don't have any numbers over here, so I just say decreases. From 8 to 10, you could say it decreases at a pretty consistent rate. That's not really a straight line, but it's kind of. So like it decreases from 8 to 10, should be all you need. And why did that happen? Oh, let's see what we got. Oh, I'm sorry, I'm on the back one. So remember, rate is always change in y over change in x. Huh? Yeah, good. It looks like I've got an enzyme. Enzymes are pretty much always proteins. And so I've got a protein and it looks like it functions well at a pretty neutral pH like 7. Looks like it's doing all right between 6 and 8. And it looks like as you start to go much below like 6, it denatures. And as you go much above 8, it's denatured. So I could say it denatured, and then I'd want to flesh that out a little bit and say something like, as the pH increases, the the shape of the enzyme changes and it can no longer do its job, and we call that denaturation, something like that. But yep, it denatured. Some students start getting this mixed up with like exothermic, endothermic graphs, and they start talking about like activation energy and stuff. But pH is down here. Exothermic, endothermic, this is time. So that's pH. All right, we are done for today. Um, don't forget, if you want to turn in your AP Bio Lab a little bit early next time, then you can get +3 bonus points. It's not officially due until the 28th, but if you turn it in by the 26th, you get +3 for turning it in early. All right. We'll check and see what we got on TCTV. If you're on the Zoom and you're going to watch TCTV on your own so you don't have to listen through my crappy speakers, then you're free to go. Yes. So rate, there's a few different ways you can say it. Let me go back. So it's just the slope, rate of slope. So anytime there's something changing over time, that slope is the rate. So some teachers said, like in a physics class, they'll say something more complicated like y2 minus y1 over x2 minus x1. Or they might say change in y over change in x. And then the simplest way to look at it is rise over run. All of these are correct. So for example, on this one, this is rise, this is run, this is y, this is x. So change in y over change in x, change in y or rise over change in run, x. So how far did I rise when I'm at temperature 10 and temperature 20? How far up did I go? Well, I went from 80 to 120. So my total rise was 40. How far did I run? Well, I went from temperature 10 to temperature 20. So my total run was 10. And you can write it a few different ways. You know, I did 120 minus 80, my change in y. I did 20 minus 10. I went from here over to here. So 20 minus 10 to get 4. But yeah, it's however you learn slope, it's slope. Yeah. So rate is slope. Next time somebody asks, I'll just start with that. I'll say rate is slope. And if they're like, okay, cool, then I can just, yeah, cool. So I should have started with that instead of ending with that. Okay, let's see what we got for TCTV. Let me get my audio hooked up.